Hello students. In today's module, we'll be studying secondary active transporters. To move substances against a concentration or electrochemical gradient, the cell must use energy. Two mechanisms exist for the transport of small molecular weight materials and small molecules across cell membranes. Primary active transport moves ions across a membrane and creates a difference in charge across that membrane, which is directly dependent on energy released on hydrolysis of ATP. Secondary active transport involves the movement of material due to the electrochemical gradient established by the primary active transport and does not directly require ATP. Secondary active transporters, which are membrane proteins, form the largest and the most diverse group of membrane proteins transporting substrates across the membrane. They are involved in nutrient uptake, maintenance of ion gradients and voltages, export of toxins, antibiotics, and proteins. They are present in all organisms, including bacteria, plant, and animals. And in this module, we'll be studying the mechanism of secondary active transport, the types of secondary active transporters, and some examples of them which are physiologically important. So what is secondary active transport? Secondary active transport is a type of active transport across a biological membrane in which a transport protein couples the movement of an ion, typically a sodium ion or a hydrogen ion, down the electrochemical gradient to the movement of another ion or molecule against a concentration or electrochemical gradient. The ion moving down its electrochemical gradient is referred to as the driving ion. The ion or the molecule being transported against a chemical or electrochemical gradient is referred to as the driven ion or molecule. This transport process is referred to as active transport because a driven ion or molecule is transported against a concentration or electrochemical gradient. It is also referred to as secondary active transport because no ATP hydrolysis is involved in this process as opposed to primary active transport. The energy required to drive transport resides in the transmembrane electrochemical gradient of the driving ion. An important feature of such co-transport is that neither substance can move alone. Movement of both substances is obligatory or coupled. So, secondary active transport is also referred to as ion-coupled transport. Those utilizing sodium ion as a driving ion are called sodium-coupled transporters. And those utilizing hydrogen ion as a driving ion are called hydrogen-coupled transporters. Two types of secondary active transport exist, symport and antiport, and the transport proteins are known as symporters and antiporters respectively. These co-transporters share common features with uniporters such as glued proteins that transport glucose. They share certain structural similarities, operate at equivalent rates, and undergo cyclical conformational changes during transport of their substrates. They differ in that uniporters can only accelerate thermodynamically favorable transport down a concentration gradient, whereas co-transporters can harness the energy released when one substance moves down the concentration gradient to drive the movement of another substance against its concentration gradient. Let's look at symport. In symport, the direction of transport is the same for both the driving ion and the driven ion or molecule into the cell or out of the cell. Mammalian cells express many types of sodium-coupled symporters. The human genome encodes literally hundreds of different types of transport proteins that use the energy stored across the plasma membrane in the sodium ion concentration gradient and in the inside negative electric potential across the membrane to transport a wide variety of molecules into cells against their concentration gradients. However, not all symporters use sodium ion as a driving ion. Some use an existing proton electrochemical gradient as well. So, a good example of symporter is a sodium-glucose cotransporter, which couples the movement of sodium ion into the cell down the electrochemical gradient to the movement of glucose into the cell against its concentration gradient. Sodium-glucose symporters. Many cells, such as those lining the small intestine and the kidney tubules, need to concentrate glucose against a very large concentration gradient. Such cells utilize a 2-sodium, 1-glucose symporter, a protein that couples transmembrane movement of one glucose molecule to the transport of 2 sodium ions. By transporting 2 sodium ions per glucose, this sodium-glucose symport protein can accumulate glucose against a much steeper concentration gradient than if only 1 sodium ion were transported per glucose. In addition to glucose, 2-sodium, 1-glucose symporter recognizes galactose as a substrate. Its function is crucial to dietary glucose and galactose absorption in the small intestine and reabsorption of filtered glucose and galactose in the proximal tubule of kidney nephrons. Cells use similar sodium ion-powered symporters to transport substances other than glucose into the cell against high concentration gradients. Several types of sodium amino acid symporters allow cells to import many amino acids into the cell. Let's look at some other examples of sodium ion-powered symporters found in many different cells and tissues performing a variety of important physiological functions. The sodium phosphate cotransporter is found in the apical membrane of epithelial cells of the small intestine and renal proximal tubules. It utilizes the sodium ion electrochemical gradient to drive the uphill transport of inorganic phosphate into the cell. For every transport cycle, the sodium phosphate couples the inward translocation of 3 sodium ions and 1 divalent phosphate ion. The sodium iodide symporter is responsible for the accumulation of iodide by the thyroid gland. It is localized to the basolateral membrane of thyroid follicular cells and co-translocates 2 sodium ions and 1 iodide ion per transport cycle. The sodium iodide symporter is also found in other tissues such as in the mammary glands, where it functions to transport iodide into the nursing mother's milk. The sodium-potassium chloride cotransporter. The sodium chloride cotransporter as well as the potassium chloride cotransporter belong to the same family of transporters. The sodium-potassium chloride cotransporter utilizes the sodium electrochemical gradient to drive the inward translocation of sodium ions, potassium ions, and chloride ions with a 1 sodium, 1 potassium, 2 chloride stoichiometry. Among other places, the sodium-potassium chloride cotransporter is found in the apical membrane of the thick ascending limb of the loop of Henle, where it performs an important role in urine concentration. The sodium chloride cotransporter utilizes the sodium ion electrochemical gradient to drive the transport of one chloride ion per transport of one sodium ion. The sodium chloride cotransporter is found in the apical membranes of the epithelial cells of kidney nephron distal tubules. Potassium chloride cotransporter co-translocates one potassium ion and one chloride ion out of the cell per transport cycle. No three-dimensional structure for any mammalian symporter has yet been determined, but structures of several homologous bacterial sodium substrate symporters have provided substantial information about symport function. For example, the bacterial sodium leucine symporter has 12 membrane-spanning alpha helices. Two of the helices have non-helical segments in the middle of the membrane that form part of the leucine binding site. Amino acid residues involved in the binding of the leucine and two sodium ions are located in the membrane spanning segment and are close together in the three-dimensional space. This demonstrates that the coupling of amino acid and ion transport in these transporters is due to direct or nearly direct physical interactions of the substrates. Now let's study the antiporters. In antiport, the driving ion and the driven ion or molecule are transported across the biological membrane in the opposite directions. Transport proteins that are involved in this type of transport are referred to as exchangers or antiporters. The plasma membrane of most cells contain one or more types of antiporters, which couple movement of a co-transported ion, often sodium ion, down its electrochemical gradient to movement of a different molecule in the opposite direction against a concentration gradient. Let's look at the sodium-calcium antiporter. An example is a sodium-calcium antiporter, which couples the movement of three sodium ions into the cell down its electrochemical gradient to the movement of one calcium ion out of the cell against its electrochemical gradient. In cardiac muscle cells, for example, a sodium-calcium antiporter, rather than a plasma membrane calcium ATPase, plays the principal role in maintaining a low concentration of calcium ions in the cytosol. Just as in other muscle cells, a rise in the intracellular calcium ion concentration in cardiac muscle triggers contraction. Thus, the operation of the sodium-calcium antiporter lowers the cytosolic concentration of calcium ions and reduces the strength of heart muscle contraction. The sodium-potassium ATPase in the plasma membrane of cardiac cells, as in other body cells, creates a sodium ion concentration gradient used to power the export of calcium ions. The drugs wabane and digoxin increase the force of heart muscle contraction and are widely used in the treatment of congestive heart failure. The primary effect of these drugs is to inhibit the sodium-potassium ATPase, thereby raising the intracellular sodium ion concentration and lowering the intracellular potassium ion concentration. Because the sodium-calcium antiporter functions less efficiently with a lower sodium ion concentration gradient, fewer calcium ions are exported and the intracellular calcium ion concentration increases. This increase causes the muscle to contract more strongly. The sodium-hydrogen exchanger, a ubiquitously found exchanger that plays a key role in the regulation of cytoplasmic pH, is a sodium-hydrogen exchanger. Here again, the sodium ion electrochemical gradient is utilized by the antiporter to extrude hydrogen ions from the cell with a 1 sodium ion is to 1 hydrogen ion coupling, thus raising the cytosolic pH. The chloride bicarbonate exchanger or the anion exchanger. Under certain circumstances, the cytosolic pH can rise beyond the normal 7.2 to 7.5. To cope with excess of hydroxyl ions associated with elevated pH, many animal cells utilize an anion antiporter, the chloride bicarbonate exchanger or AE. In addition to cation antiporters, which transport only positive ions, many cells also contain anion transporters which transport only negative ions. An important example is AE1 protein, which is the predominant integral protein of the mammalian erythrocyte. This anion antiporter catalyzes the 1-for-1 exchange of a chloride ion and bicarbonate ion across the plasma membrane. Since one singly charged negative ion is exchanged for another, there is no net movement of electric charge and the reaction is not affected by the membrane potential. Thus, the direction of the reaction is dependent only on the concentration gradients of the transported ions. These exchanges belong to the same family as a sodium-coupled bicarbonate symporters. The direction of transport by the chloride bicarbonate exchanger is governed by the concentration gradients of the chloride and bicarbonate. For example, in red blood cells traveling in the systemic capillaries, excess production of bicarbonate in the cytoplasm as a result of carbon dioxide hydration reaction catalyzed by carbonic anhydrase drives the outward translocation of bicarbonate ion in exchange for the inward translocation of chloride ion. When the red cells pass through the lung capillaries, the direction of transport is reversed, such that inward translocation of bicarbonate is coupled to the outward translocation of chloride. The bicarbonate that enters the cells ultimately gives rise to carbon dioxide, which is catalyzed by carbonic anhydrase, which then leaves the cell and ultimately the capillaries to enter the air-filled alveoli of the lungs, where it is then exhaled into the atmosphere. Secondary active transport in plants. The lumen of plant vacuoles is much more acidic. The pH is around 3 to 6 than in the cytosol, where the pH is around 7.5. The vacuolar membrane contains a V-class ATP-powered pump and a unique pyrophosphate-powered pump, both of which function to pump protons into the vacuolar lumen against a concentration gradient. The vacuolar membrane also contains chloride ions and nitrate ion channels that transport these anions from the cytosol into the vacuole. Entry of these anions against their concentration gradients is driven by the inside positive potential generated by the proton pumps or the hydrogen ion pumps. A proton sucrose antiporter in the vacuolar membranes operates to accumulate sucrose in plant vacuoles. The inward movement of sucrose is powered by the outward movement of hydrogen ions. Uptake of calcium and sodium ions into the vacuole from the cytosol against their concentration gradients is similarly mediated by proton antiporters. So now we have come to the end of today's module. To conclude, secondary active transporters are present in all organisms and form the largest and most diverse group of membrane proteins transporting substrates across the membrane. These proteins use energy stored in the electrochemical gradient of sodium ions or hydrogen ions to power the uphill movement of another substance, and so is also called ion-coupled transport. When the transported molecule and the co-transported molecule or ion move in the same direction, the process is called symport. When they move in opposite directions, the process is called antiport, and the corresponding transporters, symporters and antiporters respectively.